Okay, so today, as the name of the chapter implies, we're going to be talking about cells, how we go about studying cells. We're going to be talking about, not in this lecture, but soon we're going to talk about the difference between prokaryotic and eukaryotic cells. But yeah, so this week is mostly just about cells. So here are three different images of cells, and the first thing I want you to notice, apart from if you're just looking at these, not really knowing much about them, there's very huge differences in color. But I want you to notice the differences in size. So these are nasal cells over here, and they look somewhat similar, probably to the cheek cells that you guys looked at last week. But notice this line represents 10 micrometers. Okay, and so each of these dark circles is a nuclei or a nucleus of one of these cells. And so these are reasonably small cells. They're not huge, but they're not tiny, but they're pretty small cells. Over here we have onion cells, which again you guys hopefully looked at last week. You'll notice their nuclei here, their cell walls here, forming this brick wall-like structure that plant cells tend to do. This is scaled at 50 micrometers. So these onion cells are larger, certainly, than these cells over here. On the far right here, we have prokaryotic, and I'm going to get into that later. Prokaryotic. These are prokaryotic versus these two, which are eukaryotic. We'll notice these tiny prokaryotic bacterial cells, and you'll see that this is scaled at 500 nanometers. So 1 micrometer, one tiny micrometer over here is equal to 1,000 nanometers. Okay, so just so that you have the scale of this in your head. This one was also taken with an electron microscope, which we're going to talk about. These were taken with light microscopes. And yeah, these are just much tinier. So the point I'm trying to get home here is that we have different ways of studying cells using different techniques and different kinds of microscopy. And cells are also very, very different in their size depending on what kind of organism you're looking at. But as we're going to learn at the end of the chapter, cells are generally always small. You don't really wind up having very, very, very large cells, and we're going to talk about why. So here are two different kinds of light microscopes. This one on the left is very similar to the ones that you guys have at home. It's got your eyepieces, which I've been calling oculars. It's got your objectives. It's got your stage clip here, your coarse focus, your fine focus, your light source. There's a rheostat down here that they didn't actually tell you anything about, but you guys should know the basic components of this microscope by now, hopefully. And this particular microscope allows you to see through an object. So objects generally have to be transparent in order for you to be able to see them on this microscope here. On this microscope, this is called a dissecting microscope. And this microscope allows you to see the surface of things. Unfortunately, we don't get to use one this semester, but that's okay. It's usually just like 20 minutes in lab that we do dissecting scopes for. This is great for looking at the surfaces of rocks or bugs or whatever it is that you want to be able to see, but you can't necessarily see through. So compound scope over here. I don't know if I said that or not, but this is a compound scope. This is a dissecting scope. You see through things with this one. You see the surfaces of things with this one. And dissecting scopes, generally less magnification than compound scopes. The next sort of level of distinction is that this is a compound scope, again, like the ones that you guys have at home. This is looking at a bunch of bacterial cells. This is an electron microscope, which obviously is very, very much closer. You get these magnificent images of things. This is colorized, by the way. So they've taken multiple images and then they've added color to them based on, what they can see with a light microscope, what they know the colors to be. These things are hundreds of thousands of dollars, okay? These things are a few $100, which is not cheap, certainly. For a good one, you can get into a few $1,000s, but electron microscopes, super, super expensive, and I'm sure you can see why. You can get these really very fantastic, super close images of things. They actually have an electron microscope up at the University of Pacific. Super cool. I never got to use it. My project just didn't take me there, but I always wished that I had been able to. Anywho, this is a light microscope. This is an electron microscope. You should know electron microscopes focus a beam of electrons, which gives you, it allows you to get much, much closer to things, okay, than just a regular light microscope. The book it emphasizes certain careers, and in this career that it's emphasizing is a cytotechnologist. And what this is just saying is basically that we can use these ideas of cells and our ability to study cells within the context of health. Okay, so a cytotechnologist is somebody who has a bachelor's degree. Generally, they're not a doctor. They're just staining and resolving cells. So this is from a pap smear. Okay, these are normal cells on the left, and the ones on the right are infected with HPV. And the reason I know that is because the book told me. I'm not personally a cytotechnologist myself. But yeah, the point here is that you can use microscopy, you can study our cells, and there are valid applications within the field of health. Okay. So this is the generalized structure of what's referred to as a prokaryotic cell. Okay, that's one to write down. Prokaryotic cell. A prokaryotic cell does not have a nucleus. A prokaryotic cell, I'll say it again because you need to know it. A prokaryotic cell does not have a nucleus or membrane-bound organelles. And I'll define that later, don't worry, but prokaryotic cell does not have a nucleus or membrane-bound organelles. But let's look at what it does have. It has these pili here, singular pilus, and these things can be used for these single-celled organisms to basically have sex or exchange genetic information with one another. A pilus can also be used, or pili can be used to help anchor it to certain surfaces. And these are on the very outer perimeter, the very border of these cells. The most striking thing here that it has is a flagellum or a flagella, and flagella are used to make this thing move. It is for motility fundamentally. Which is just, again, a fancy way of saying this thing is used like a little propeller that makes the cell move. It has a capsule, which you're not really going to see in eukaryotic cells. It has a cell wall, which you will see in eukaryotic cells. And it has a cell membrane, which you will see in all cells. All cells have a cell membrane. It has these ribosomes, which are not organelles. They seem like they're organelles, but they're not true organelles. And we're going to talk about what these things do in a later lecture. But the long and short of it is ribosomes are used to make protein. And we talked last week about how important protein is for all kinds of stuff. Okay. The cell also has this chromosomal DNA that's just kind of hanging out in the middle. Okay, it has this chromosomal DNA that just hangs out in the center. No nucleus to contain it. The DNA just hangs out in the middle. Okay, and the region's called the nucleoid. And yeah, so if it was eukaryotic, it would have what? Yeah, actually, if you shoot me an email that subject line is prokaryotes versus eukaryotes, and there's probably going to be more than one of these, but if you say prokaryotes versus eukaryotes, and you tell me some of the things that a prokaryote would have that a eukaryote wouldn't, you'll get some extra credit. So here is the size. Move this a little bit. Here's the relative size of some things. So if you go down to 0.1 nanometers, very, very, very tiny, you see we have an atom. Okay, from the chemistry section last week. As we increase, we get an order of magnitude larger, and we start having lipids. Okay, remember those are fats from last week. We get almost an order of magnitude larger. We run into the proteins. Now, proteins can be all sorts of different sizes. They can wind up being very large or sometimes quite small, but this is just a generalization. Then we start getting toward the edge of what we're actually able to resolve with regular light microscopes. So this is not an electron microscope, but we can see down to about 100 nanometers. It's the size of a bacteria. Bacteria actually are range in size very, very widely, but this is a generalized size. Bacteria, by the way, as maybe you figured out from the previous slide, are prokaryotes. You and I are eukaryotes. Sorry for the pun, but that's how I always remember it. Eukaryote, you. We get a little bit larger and we run into these mitochondria, which there's a pretty great story about mitochondria and endosymbiotic theory that I'll maybe make another video about later on. But inside of animal and plant cells, you have these mitochondria. Okay? Mitochondria live inside all of these. We get larger, we get to animal and plant cells, which you guys all looked at under the microscope, hopefully last week. As we continue getting larger, we run into a human egg cell. We continue getting larger, we run into a frog egg cell, a chicken egg cell, an ostrich egg cell, and finally we get to an adult female. I don't know why they put an adult female at less than 1 meter tall, but that's okay. Kind of weird, but that's fine. This just gives you a sense of the relative sizes of different kinds of cells. So I would definitely, if I were you, I would know that prokaryotic cells in general are much smaller than eukaryotic cells. Prokaryotic cells are much smaller than eukaryotic cells. And I think, I think that's all I'm going to do for this lecture.